Biomolecules and the cell, Satyanarayana Chapter 1. 90% body weight is composed of carbon, hydrogen, oxygen, nitrogen, phosphorus. 90% of compounds are carbon containing. Escherichia coli has 6,000 organic compounds, whereas human has 1 lakh organic compounds. These are the estimated values. Then lipids. They're not biomolecules in a strict sense, but majority of them contain fatty acids. In a chemical composition, like water is a solvent of life, and a 65 kg man, lipid, this is protein is majority component, lipid is mostly the adipose tissue, and carbohydrate is of low content, which is in the form of glycogen. Now next is the cell. Cell is a basic unit of life, proposed by Schleiden and Schwann in 1836. Then majority of percentage is of water in the body. So water is a majority solvent component, which is nearly 60%. And if we take this 65% kg of a man to be the ideal value, then the weight of water is approximately 40 kgs in a man. Next. After water, it is proteins, which is in the muscles. And next is lipid. Protein is 17%, protein is 17%, which comes after water, that approximately 11 kgs. Next is lipid, that is 13.8%, 9 kgs, carbohydrate 1.5%, 1 kg, and minerals are 6.1%, 4 kg. Now prokaryotic and eukaryotic cells. Prokaryotic, as we know, lacks a defined structure. Eukaryotic is well-defined structure. Now eukaryotic cell, in eukaryotic cell, in human body, there eukaryotic cell, there are approximately 10 power 14 cells, and the cell size is approx 10 to 100 micrometer. And in plant cell, the plant cell differs from animal cell by possessing a rigid cell wall mostly composed of cellulose and chloroplast. The latter is the site of photosynthesis. Now nucleus. Nucleus is about 90, the diameter of nucleus. Diameter is about 90 nanometer. That is, two nuclear membranes have nuclear pores. Two nuclear membrane. For example, if this is the nucleus, it has two membranes. So the two nuclear membranes having nuclear pores. These have some nuclear pores. The nuclear membrane has some nuclear pores in between. These are the nuclear pores. Nuclear pores, and with diameter of about 90 nanometer. They have a diameter of about 90 nanometer. These pores permit the free passage of products synthesized in the nucleus into the surrounding cytoplasm. So the two nuclear membranes have nuclear pores with a diameter of about 90 nanometer. Next is the medical or clinical concepts. First is lipofuscin. Lipofuscin is a pigment rich in lipids and proteins, and it is implicated in aging process. So rich in lipids and proteins. Rich in lipids and proteins, and it is implicated, helps in the aging process. Next is leakage of lysosomal enzymes into cell degrades several functions of macromolecules. Arthritis. Why is arthritis caused? Arthritis is caused due to the leakage of lysosomal enzymes. So arthritis is caused due to the leakage of lysosomal enzymes into the cells that degrade several functions of macromolecules, and this may lead to certain disorder, that is arthritis. So we can see the image of arthritis. Arthritis. So this is the example of arthritis. You can see here that leakage of lysosomal membranes has caused this. Instead of this, we can take a realistic image, that is This would be better. So this is a picture of arthritis. Next, Zellweger syndrome. So why is arthritis caused? Leakage of lysosomal enzymes into the cell degrades several functional macromolecules, and this may lead to certain disorders. Now next is Zellweger syndrome. Next is Zellweger syndrome. Zellweger syndrome is a rare disease characterized by the absence of functional peroxisome. The peroxisomes are absent. Functional peroxisomes are absent. That is Zellweger syndrome. It's a rare disease. We'll see how it is. Zellweger syndrome. This is the Zellweger syndrome. Zellweger syndrome. This is Zellweger syndrome, that is, peroxisomes are absent. Next, is the defect of cytoskeletal protein, namely spectrin in RBC, result in hereditary spectrocytosis. This result in breakdown of RBC, which is anemia. So now we'll be learning about anemia. Anemia is caused because of defect in cytoskeletal protein, namely spectrin of RBC. So the spectrin of RBC results in hereditary spherocytosis. Hereditary spherocytosis, and this results in breakdown of RBC. This results in breakdown of RBC. So the anemia spectrum of RBC, which results in hereditary spectrosis, which results in breakdown of RBC. Go to the Google. Anemia. See, breakdown of the RBCs. This is how it occurs. Breakdown of RBCs. Biconcave shape of the RBCs is broken down, so that is called anemia spectrin. So defect of cytoskeletal protein, namely spectrin. So spectrin in RBC is nothing but cytoskeletal protein. Cytoskeletal protein, namely spectrin in RBC, is a cytoskeletal protein. Next, nucleosomes. In DNA, in eukaryotic DNA, the basic protein histones are in the ratio 1:1 to form nucleosomes. An assembly of nucleosome constitutes chromatin fibers called chromosomes. Next is Hutchinson-Gilford, HGPS. This is Hutchinson-Gilford progeria syndrome. Hutchinson-Gilford progeria syndrome. It is a rare condition of aging beginning at the birth. So this is aging beginning at the birth. Aging beginning at birth, and this is a rare syndrome wherein 1 birth out of 5 million births gets this SGPS. And why this SGPS is called? Because of the distortion of nuclear envelope due to accumulation. Distortion of nuclear envelope due to accumulation of abnormal protein. Lamin A is a abnormal protein. Lamin A is abnormal. Lamin A is an abnormal protein. So Hutchinson-Gilford progeria syndrome. HGPS. This is HGPS. Now, next is mitochondria. We'll be learning the dimensions of mitochondria. Mitochondria. It's a filamentous rod-like structure and the dimensions are 1.0 into 3 micrometer, and there are about 2000 mitochondria, and it occupies about one-fifth of the total cell volume, in a typical cell. And the mitochondria, it contains of two membranes. The outer layer is smooth, and the inner layer is called a cristae, and this is called the matrix of the mitochondria. Now, the crests which occupy the cristae is formed due to to occupy more surface area, larger surface area. And the matrix contains several enzymes concerned with the energy metabolism of carbohydrates, lipids, and amino acids. Several enzymes, that is citric acid cycle, beta oxidation. The matrix enzymes also participate in the synthesis of heme and urea. So the matrix, it first of all, it has several enzymes concerned with the energy metabolism of carbohydrates, lipids, and it is also present in the synthesis of heme and urea. Matrix contains, these are all enzymes. Next, the 10%. Now the mitochondrial RNA is called as, the mitochondrial DNA is called mtDNA. It is denoted like this. It is circular and double stranded. That is mtDNA. It also contains RNA as well as ribosomes. So it has an independent protein synthesizing machinery, and it is known that 10% of proteins is synthesized by mitochondria alone. The structure and disciplines of mitochondria is closely resembled to it closely resembles prokaryotic cells. It closely resembles to prokaryotic cell in function. That's why it is believed that during evolution there are two organisms. That is, one was the aerobic bacteria. Since bacteria is a prokaryote, we can take aerobic bacteria plus anaerobic eukaryote, and these together, this aerobic and this eukaryote together were in the evolution they formed aerobic eukaryotes, which in present day is called a eukaryotic cell. Aerobic eukaryote. Next, endoplasmic reticulum. Now, the important point of endoplasmic reticulum, it contains two types, that is rough RER and SER. RER is rough because of the presence of ribosomes which help in protein machinery. These all things are known by us. And SER helps in the synthesis of lipids, that is triacylglycerols, phospholipids, steroids, and metabolism of drugs, besides supplying the calcium for cellular functions. That is, the sarcoplasmic reticulum supplies calcium for the cellular functions. During the process of the cell fractionation, rough endoplasmic reticulum is disrupted to small vesicles such as microsomes. So what is it? During cell fractionation, fractionation, rough ER, rough endoplasmic reticulum, is disrupted to form small vesicles known as microsomes. Microsomes, but these microsomes are usually, they do not occur in cell. Do not occur. They usually do not occur in the cell. So next is Golgi apparatus. So the Golgi apparatus contains parallel stacks or vesicles, membrane vesicles known as dictyosomes. Dictyosomes. And Golgi apparatus catalyze the addition of carbohydrates, lipids, or sulfate moieties to the proteins. These chemical modifications are necessary for the transport of membrane across the plasma membrane. Certain proteins and enzymes are enclosed in membrane vesicles. So there are certain cells, and if it gets the stimulus, then only the enzyme is released out, the protein enzyme. This is the function of pancreas, digestive enzymes of pancreas. So certain proteins and enzymes are enclosed in membrane vesicles of Golgi apparatus. Certain proteins and enzymes are enclosed in membrane of Golgi apparatus and secreted from the cell after appropriate signals. So this is the digestive enzyme of pancreas produced in this fashion. Now Golgi apparatus also catalyze the addition of carbohydrates, lipids, and sulfate moieties. These connect them to the proteins, so that after these are connected, then only the protein, then if this is a cell, then only the protein can exit out of the cell. That is, these modifications by the Golgi apparatus, that is adding carbohydrates, lipids, sulfate, are necessary for the transport of proteins across the plasma membrane. Now this pancreas is told. Now Golgi apparatus has three functions. First of all is the modification, that is catalyze the protein so that they go out of the membrane. Second one is the pancreas, that is after the stimulation only the enzymes and proteins are released out of the cell, and this is the mechanism for digestion of pancreas. Now third function is membrane synthesis. Third function is membrane synthesis of the intracellular organelles like the peroxisomes and lysosomes. Next, lysosomes enzymes are called hydrolases, and the important point is these include enzymes with substrate in the background. Alpha glucosidase is for glycogen. What we need to remember is cathepsins is for proteins. The lysosomal enzymes are responsible for maintaining the cellular compounds in a dynamic state. Now there's another called inclusion cell, that is eye cell disease. Inclusion cell disease is a rare condition due to absence. This is due to absence of certain hydrolytic hydrolases in the lysosome. Inclusion cell eye disease is a rare condition due to absence of certain hydrolases in lysosome. So a defect in protein targeting, as the enzymes cannot reach the lysosomes. Eye cell disease. This is the pic of eye cell disease, that is inclusion cell eye disease is a rare condition due to absence of certain hydrolases in the lysosome. However, this enzyme is synthesis and found in the circulation. Eye cell disease is due to a defect in protein targeting, as the enzymes cannot reach lysosomes. Next is peroxisomes. Peroxisomes has three functions. It first of all has enzyme catalase. Catalase has three functions. First function is it converts toxic H2O2 to water and oxygen. Second is it does the work of oxidation of long chain fatty acids. That is more than 18 carbons in the chain. And third is it does synthesis of plasmalogens and glycolipids. Plants contain, now in addition to this, plants contain glyoxysomes. It's a specialized type of peroxisome which are involved in glyoxylate pathway. This is involved in glyoxylate pathway. Next, this we're going to study about PBDs. That is peroxisome biogenesis disorder. Peroxisome biogenesis disorder are a group of rare diseases involving the enzyme activities of peroxisome. The biochemical abnormalities associated with PBDs includes increased levels of, so what happens in PBDs is increased levels of very long chain fatty acids, that is C24 and C26, and decreased concentration of plasmalogens. The most common of this form is Zellweger. This itself is the Zellweger syndrome. This one. Peroxisomes absent, Zellweger syndrome. PBDs. Zellweger syndrome, this is itself PBDs. This is a Zellweger syndrome, peroxisome absent, PBDs. Next. And important point is the victims of this disease may die within 1 year after birth. So victims die within 1 year after birth. Zellweger syndrome victims. Cytoskeleton. The cytofilaments are three types, that is microtubules, actin filaments, and intermediate filaments. Last point is the cytoskeleton contains of cytoplasmic filaments that are of three types. One is microtubules, another is actin filaments, next is intermediate filaments. Last is apoptosis. Apoptosis is nothing but the natural cell death. Thank you.